So, hello everyone. So, we are third year veterinary medicine students from Block B of De La Salle Araneta University, and this is our pathology lecture, final product discussion. So, I am Regine Chloe I. Noriega.
And I am Jeremy E. Oranga.
And I am Mark Jantemenya.
Okay. So, first we will be discussing the cell and tissue injury.
Yes. Okay, guys. Let's start with the aims of the topic. Number one is to introduce the concept of structural and functional cell changes in response to the harmful or injurious stimuli as the basis of gross pathological lesions. Number two is to define cell changes as irreversible, for instance, adaptation and degeneration, or irreversible, such as death, necrosis, and to discuss commonly encountered examples of each. Last one is to discuss some common extracellular changes in tissues in response to various stimuli. Okay, let's talk about lesions. Lesions are pathological changes in tissues or organs. There are two types of lesions. Number one is the gross lesions, abnormalities that we can see in our naked eye. Manifestations of abnormalities or changes in the cell that make up the tissue or organ. Second is the cellular lesions, which basically is the changes or abnormalities in the cellular level. Okay. Additional info, if abnormalities occur at the cellular level, they are most likely lead to changes in tissue structure. Another effect is that it may alter the function of the tissue. Next, let's talk about injury following harmful stimulus. Number one, first, yung reversible cell injury. Cell injury can cause alteration or loss of cell function and structural changes, but the cell can still recover and regain structure and function if the injurious stimuli has been removed. It is also called the sublethal cell injuries or changes. Basically, ang reversible cell injury is may loss of cell function pero nababalik naman ito. Nakaka-recover sila. Okay, second, irreversible cell injury. Cell injury can cause alteration or loss of cell function, same with the first one, and structural changes, but the cell would not have the ability to recover even if the stimulus is stopped or removed. Ito naman yung hindi na nagre-recover, irreversible cell injury. Next, let's go to the cell response following injurious or harmful stimuli. First, the degeneration. Basically, a reversible response. Nababalik. Second, necrosis, an irreversible response and the cell dies. Necrosis means death. Okay? Changes in cell, number three, changes in cell growth, size, number. These can be either reversible or reversible. Next, factors affecting the type of cellular damage which occurs in response to harmful stimulus. Number one, factors specific to the injurious stimulus. Duration. The period of time, how long the harmful stimulus goes on for. Number two, size. How large the harmful stimulus is. Kung gano'n lang siya kalaki, basic lang. Number two, sorry, dun sa numbering ko, nagkamali ako. Factors specific to the target tissue. First, blood supply. Kung how well supplied yung blood tissue. Secondly, yung metabolic characteristics, how metabolically active the tissue is. Okay, next topic. I'll give it to Mark Jantimen.
Second is degeneration and necrosis. First off is degeneration. Degeneration is a reversible cell injury. The injured cell can regain homeostasis and return to a morphologically normal state. The affected organ or tissue will become swollen and discolored because so many of the cells making up the organ are swollen with the accumulated substance. Next is accumulation of substances. First off is cellular components, accumulation of cellular components. As cells age, old and non-functional organelles may shrivel up or be broken down, but their constituents may remain in the cytoplasm. Uh, they tend to accumulate as fatty, protein, or crystal deposits and can often be seen when the cells are examined under the microscope. Second is hydrophic change, which is cloudy swelling. Damage to cell membranes means that cells swell with fluid, occurs in many cell types, example of early stages of liver cell damage by toxins. Next is fatty change, or the fat accumulates in cells because of increased or long-term fat breakdown in the body, and thus the cells encounter increased or long-term levels of fat in their immediate environment. This happens in malnutrition, when body fat is broken down and carried to the liver for metabolism because there are inadequate dietary sources of energy. The liver cells therefore are presented with large amount of fat in the blood and entering the liver. Fatty change can also occur because the ability of cells to metabolize normal amounts of fat is decreased by cell damage. Decreased cell metabolism because of toxic damage or reduced oxygen supply due to heart dysfunction, respiratory disease, or anemia, or in diabetes mellitus. Fatty change can be idiopathic or uncertain cause in the liver of older cats. Next is pigments. Accumulated pigments may be endogenous, produced by the body, such as hemosiderin, breakdown product of red blood cells, which may accumulate if there is marked pigments accumulate in disorders which cause jaundice, such as liver disease. Melanin is the brown pigment in skin and may accumulate in skin cells in certain conditions such as chronic dermatitis, causing the skin to darken. Accumulated pigments may also be exogenous or foreign to the body, um, foreign to the body, such as carbon from pollution, which may accumulate in the lungs of animals living in built-up areas. Other exogenous pigments can also accumulate in cells. Indeed, we make use of the fact since the inks used in tattoos accumulate in the cell skin. Next is proteins. Viruses have to use cells of living creatures to manufacture protein, as they do not have the capacity to do so for themselves. Without using cells this way, viruses would not be able to reproduce themselves because of this in some viral disease. Viral proteins accumulate within the cells and can be seen under the microscope, so-called viral inclusions. Uh, some viral inclusions are very characteristic in appearance and can help to make a diagnosis of a specific viral infection even before virology has been carried out. Next is the forms of degeneration. First off is the cell swelling, an early change that occurs in most types of acute injury and which may be a prelude to more drastic changes. Next is ballooning degeneration or balloon degeneration. Sorry, di nakikita siya sa screen. Um, Caused by membrane damage, which allows influx of fluid into the cell, or by damage to cytoskeleton, leading to leading to loss of cell shape. Next is hydrophy degeneration, which is ion and fluid homeostasis that lead to an increase of intracellular water. Next is hyaline degeneration. Sorry, hindi nakikita uli yung ano, pangalan niya. Tissue degeneration, chiefly of connective tissues, in which structural elements of affected cells are replaced by homogeneous translucent material that stains intensely with acid stains. Next is mucoid degeneration. A rare pathological affection of the anterior cruciate ligament injury, ganglion cyst, and degenerative process have been implicated as the most likely etiological factors in the production of this change. Next is fatty degeneration, the accumulation of fat globules within the cells of an organ such as the liver or heart, resulting in deterioration of tissues and diminished functioning of the affected organ. Next is colloid degeneration, a rare cutaneous deposition disorder characterized by a single large nodule or multiple soft to rubbery flakes or nodules occurring over sun-exposed regions. Conversion of the tissues into a gelatinous or gum-like material. Lastly is the Wallerian degeneration, an active process of retrograde degeneration of the distal end of an axon that is a result of a nerve lesion. Next is the necrosis. First off, the necrosis is a irreversible injury. It means it cannot regenerate or turn back to its normal state. Increased eosinophilia, nuclear shrinkage, fragmentation, dissolution, breakdown of plasma membrane or organellar membranes, abundant myelin figures, leakage, and enzymatic digestion of cellular contents. Necrotic cell may persist for some time, may be digested by enzymes and disappear. First of necrosis that is the type of cell that is associated with the loss of membrane integrity, leakage of cellular components, culminating dissolution of cells, largely resulting from a degradative action of enzymes on lethally injured cells. The leaked cellular contents often elicit local host reaction called enzymes called inflammation that attempts to eliminate the dead cells and start subsequent repair process. The enzymes responsible for digestion of the cell may be derived from the lysosomes of the dying cell themselves and from the lysosomes of leukocytes that are recruited as part of the inflammatory reaction of the dead cells. Next is the common causes of necrosis. First off is decreases blood supply such as may occur when a blood vessel is damaged or blocked. Next is pressure, from a tumor pressing on the area or prolonged presence of a tight bandage. Next is burns. Burns may be caused by heat, extreme cold, or caustic chemicals. Next is trauma, causing damage to blood vessels or pressure on tissues. Next is poisons and toxins, kill off cells by harmful effects on metabolism. And lastly is infectious agents like bacteria, viruses, fungi, and parasites that damages cells in various ways. Next is sequelae. Di ko mabasa, pero try ko lang. Sequelae of necrosis. Ito naman yung this part is what happened next or likely the possible next stages of the necrosis. First off is the tissue regeneration, healing of the area by replacement of dead cells by cells of the same type. Next is scar formation, healing of the area by replacement of dead cells by fibrous tissue. Next is erosion or ulceration, loss of cells from a surface in or on the body, usually accompanied by inflammation. Lastly is sequestration, the area of dead and dying cells become walled off from the rest of the body by a dense rim of fibrous tissue, which means that the necrotic process, usually accompanied by inflammation, can continue inside. Next is the forms of necrosis. Medyo magulo yung pero sige, try natin. I-ano. Um, First off is the coagulative necrosis. The most common type of necrosis, and it occurs in many of the solid organs, such as the kidney and heart. Affected organs retain their structure and are pale and firm as though they had been cooked. After a period of time, inflammatory cells move into start to move to remove the dead cells. So the appearance of coagulative necrosis changes with time. Next is the caseation necrosis. In caseation necrosis, the dead tissue is converted to a cream cheese texture, hence the name caseous means cheesy. This type of necrosis is usually associated with chronic bacterial infections such as tuberculosis, and is due to the presence of special fats in the bacterial wall which prevent liquefaction taking place. Next is liquefaction necrosis. Refers to areas of necrosis which become liquefied due to release powerful enzymes which degrade the dead cells and extracellular components, leaving a thick soup. This type of necrosis is characteristic after insults to the brain, such as follows interruption of the blood supply or bacterial infection. Next is special forms of necrosis like fat necrosis. Fat necrosis occur after inflammation in or around fatty tissue, and occurs because enzymes degrade fat cells that the fat becomes hard and nodular. This is seen in the abdomen after pancreatitis when pancreatic enzyme leak into the fat into the peritoneum. Next is gangrene. Gangrene is a variant of coagulative necrosis and is the necrosis occurs specifically due to loss of blood supply to an area, especially an extremity such as foot, ear, tip, or tail. There are four forms of gangrene. There are dry gangrene, that is no bacteria are involved. The tissue is dry and shriveled, and it is discolored. Next is gas gangrene, which gas producing bacteria proliferate in the necrotic tissue. The affected tissue has a crackly, crepitant feel. Next is moist gangrene. Pus-producing bacteria proliferate in the areas of necrosis. This type of gangrene characteristically appears rotten and is very foul smelling, which is putrefactive. Next is wet gangrene. Bacteria are present and there is inflammation of adjacent non-necrotic tissue. Inflammation in connective tissue is called cellulitis. The area swells and oozes fluid. Next is infarct. Due to inadequate blood supply to the affected area, it may be caused by artery blockage, rupture, mechanical compression, or vasoconstriction. The resulting lesion is referred to as an infarct. Lastly is the Zenker's necrosis, a severe glassy or waxy hyaline degeneration or necrosis of skeletal muscles in acute infectious disease. A prototype It is a prototype of a coagulative necrosis. This condition was named by Friedrich Albert von Zenker. May I add the apoptosis? It means a pathway of cell death in which cells activate enzyme that degrade the cell own nuclear DNA and nuclear and cytoplasmic proteins. Fragments of the apoptotic cells then break off, giving the appearance that is responsible for the name apoptosis or falling off. The plasma membrane of the apoptotic cell remains intact, but the membrane is altered in such a way that the cell and its fragments become avid targets for phagocytes. Next is Causes of apoptosis. Apoptosis in physiological situations, death by apoptosis is a normal phenomenon that serves to eliminate cells that are no longer needed and to maintain a constant number of cells of various types in tissues. First, in the physiological situation is the programmed destruction of cells during embryogenesis. The normal development is associated with the death of some cells and the appearance of new cells and tissues. The term programmed cell death was originally coined to denote this death of specific cell types at defined times during the development of an organism. Next is involution of hormone-dependent tissue upon hormone deprivation, such as endometrial cell breakdown during menstrual cycle and regression of the lactating breast after weaning. Next is the cell loss in proliferating cell population, such as intestinal crypt epithelia in order to maintain a constant number. Next is the elimination of cells that have served their useful response, such as neutrophils in an acute inflammatory response and lymphocytes at the end of an immune process. In this situation, cells undergo apoptosis because they are deprived of necessary survival signals such as growth factors. Next is elimination of potential harmful self-reactive lymphocytes, either before or after they have completed their maturation in order to prevent the reaction against the body's own tissues. Lastly is the cell death induced by cytotoxic T lymphocytes, a defense mechanism against viruses and tumors that serves to kill viruses, infected, and neoplastic cells. Next is the apoptosis in pathological conditions. First off is the apoptosis eliminates cells that are genetically altered or injured beyond repair and thus so without eliciting a severe host reaction, thereby keeping the extent or tissue damage to a minimum. First off is the DNA damage. Radiation, cytotoxic, anti-cancer drug, extremes of temperature, and even hypoxia can damage DNA, either directly or through production of free radicals. If repair mechanism cannot cope with the injury, the cell triggers intrinsic mechanisms that induce apoptosis. In these situations, um, elimination of the cell may be a better alternative than risking mutation in the damaged DNA, which may progress to a malignant transformation. Next is the accumulation of misfolded proteins. Improperly folded proteins may arise because of mutations in the genes encoding these proteins or because of extrinsic factors such as damage caused by free radicals. Excessive accumulation of these proteins in the ER leads to a condition called ER stress, which culminates in apoptotic death of cells.
So next, let's discuss inflammation. It is defined as a complex progression of blood, vascular, and tissue changes that develops in response to tissue injury. Yung response na mentioned ay primarily a protective one because as stated, it extends various protective mechanisms such as phagocytic cells out into the tissues and prepares way for tissue repair. On the other hand, in veterinary approach, inflammation is often initiated and worsened by self-harm like scratching, biting, licking, etc. And one of the general knowledges about inflammation is it often works with and is directed by cells of immune system. Inflammation is described as acute and chronic depending on the duration of inflammatory response and inflammatory cells involved. The first one is acute inflammation. It is described as the initial reaction of a tissue to injury, and it aims to rapidly contain, dilute, and remove the harmful stimulus which has set off the inflammatory response and to prepare the tissues for repair. On the cardinal signs of acute inflammation, please refer to the photo on the right. Uh, the three phases of acute inflammation. The first one is the vascular. It has change in blood vessels diameter and it is subdivided into two parts. The first one is the white line. It is called the white line. The constriction in the arterial supplying the capillary bed. Mas maikling occurrence nito compared sa hyperemia. Ma-demonstrate to by scratching yourself with one finger using your nail. Pag scratch nyo, may mag-appear na white line dyan. Pag nag-appear yung white line, meaning present yung constriction na ito. Pag nag-appear ng short period. Next is the hyperemia. It is the dilation of arterial and capillary and caused by chemical mediators from damaged tissue and from plasma. It lasts longer, minsan minutes or even days, and it also causes area to become reddish and warm. The next phase of inflammation is called exudative. There's an increase in blood vessel permeability resulting in fluid leakage from the circulation. There is also an escape of protein-rich fluid from blood into surrounding damaged tissues. Maari na result ito ng direct damage sa endothelial capillaries since mga harmful stimulus that triggered the inflammatory response like physical damage, toxic agents, and etc. As you can see, the intercellular junctions are opened up. So, dyan lumalabas yung protein-rich exudates. Then, the last phase is called the cellular phase. Leukocytes leave blood vessels, travel to site of injury, and become activated. The main leukocyte in this inflammation is neutrophil. This phase is also known as period of recruitment. Sa phase na ito, nagre-recruit ng leukocytes sa site of injury, which destroy bacteria at site and mag-lead sa pag-direct ng other cells to start removing dead and dying cells para mag-start yung healing process. Okay, so now we discuss chronic inflammation. It is defined as the persistence of inflammatory response for longer than approximately 10 to 14 days. Pero based sa source nito, which is the general pathology for veterinary nurses, pwede rin maging a year or more depende sa severity. Chronic inflammation is not exudative and it involves the formation of fibrous tissue. Macrophages, lymphocytes, and plasma cells are important cells in this inflammation, accompanied by reactive hyperplasia and hypertrophy. So now let's discuss the two main types of chronic inflammation. First is the non-granulomatous. It involves loose clusters of inflammatory cells without particular arrangement and a variable background of fibrosis or with reactive hyperplasia or hypertrophy in surrounding tissues. So dahil sa presence ng hyperplasia sa skin glands ng mga chronically inflamed skin, they may become thickened and greasy. So next type is the granulomatous. It is composed of distinct arrangement of inflammatory cells called granulomas. As you can see, granulomas are made up of central collections of macrophages, along with other mononuclear inflammatory cells surrounding the macrophages and a rim of fibrous tissue outside. Okay, next topic is tissue repair. It is defined as the process by which cells lost by necrosis are replaced by living tissue. All tissues except teeth are able to heal to a certain extent. The new living tissue is not necessarily the same as the tissue it is replacing. The extent to which the process of tissue repair occurs depends on the regenerative capacity of the cells in tissue, the extent of the defect to be filled, That is the amount of necrotic tissue requiring the replacement. Uh, in this part, we discuss the two processes involved in tissue repair. First is the regeneration. Regeneration is replacement by cells of the same type. There, yung necrotic cells are already dead kaya hindi na kaya mag-regenerate. So instead, yung surrounding living cells yung magre-regenerate and mag-fill up ng gap na iniwan ng dead cells. Para mag-heal yung damaged cells damaged tissue by regeneration, the cells that make it up must be able to divide by mitosis. So next yung categorization of cells based on their ability to regenerate and multiply. The first one is the labile cells. They continue to multiply by mitosis throughout life and replace lost cells due to normal turnover. Excuse me. The next one is the stable turnovers, but not as quick as the labile cells. But following tissue injury, they rapidly divide to replace damaged cells and have longer lifespan. Permanent have no or very limited capacity to regenerate new cells. And for additional, stem cells. Stem cells are present in both labile and stable cell populations and has the capacity to differentiate according to the body or tissue requirement. So, yung next process is the organization. It is a replacement by a fibrovascular tissue or also known as scar tissue. Cells cannot regenerate if there's a large area of necrosis or cells are far from each other, and these are the reasons why the cells can fill up the gap quickly enough or not at all. As you can see in the picture, part A is where the demolition or inflammatory phase occurred. Present ang neutrophils and macrophages dito. This is where the cleaning up of the wound or damaged tissue occurs, performed by neutrophils and macrophages, and sometimes lymphocytes, plasma cells, and eosinophils are present. Then sa part B is where the granulation phase occurred due to the presence of the immature connective tissue or granulation tissue to start filling up the wound. And finally, the part C is where the maturation occurred due to the presence of fibrous scar by the process of fibrosis.
Hi, my name is Daryl Esteban, and now we're going to talk about the circulatory disturbances. In this topic, we are now going to talk about the circulatory system that is made out of the heart and blood vessels and is essentially to keep the body functioning. It is finely tuned system carry oxygen, nutrients, electrolytes, and hormones throughout the body. In this topic, we will talk about the interruption, blockage, or diseases that affect how the heart or blood vessels pump blood can cause complications. The components of the cardiovascular system, angiology, a branch of anatomy, which deals with the study of circulatory system. Circulatory system is made out of parts of the structure responsible for blood circulation and nutrient distribution throughout the body. The man in the image is William Harvey, the father of blood circulation. He is the father of blood circulation because he is the first to recognize the full circulation of the blood in the human body. So the components of the circulatory system is the heart, which has 3 layers of epicardium, myocardium, and endocardium, 4 chambers, the left atrium, right atrium, left ventricle, right ventricle, and 4 valves is the tricuspid valve, pulmonary valve, mitral valve, and aortic valve. Next is the blood vessels, which consist of arteries, arterioles, veins, venules, and capillaries. Next is the concentric layers of the blood vessel. Tunica intima, the inner layer, is made up of endothelial cells. Next is the tunica media, the middle one. Middle layer that is made of smooth muscles, and tunica adventitia, which is made of connective tissues. Next are the blood corpuscles. Blood has solid blood corpuscles, and liquid are plasma and serum components. There are 2 types of blood cells, which are erythrocytes, the red blood cells, and leukocytes, the white blood cells. In this image, we can see the white blood cell attacking the bacteria, and the surrounding circles are the red blood cells. Red blood cells are biconcave discs which contains oxygen, and white blood cells are soldiers of the body, which are, or in other terms, is fighting infections. Next here, we have a table of blood corpuscles, which are granulocytes, and granulocytes are granules that are present in cytoplasm, neutrophil, eosinophil, and basophil. Neutrophils are N cells, and it only stays less than 1 day in the blood, and nucleus of immature are slightly indented, band in other terms, or upon maturity it becomes hyperindented, meaning segmenters. Eosinophils are also N cells that last 8 to 12 days. Um, red to pink color, cytoplasmic granules reacting with eosin. Lymphocytes are not N cells, and they are multilobed nucleus and granules react with hematoxylin. Next are blood corpuscles, granulocytes. Uh, First, we're going to talk about the types of antibodies, which are the IgM, the primary response, with a pentameric structure regarded as the largest Ig. Next, IgG, the secondary response, anamnesis, or in colostrum of all animals, which are milk in animals. IgA in mucosal surfaces and secretion, which are saliva, milk, bile. IgE is in allergic reaction and parasitic infection. IgD is are found in joints, serve as antigen receptors to antibody, especially IgG. Next, we're going to talk about the granulocytes in the table, the lymphocytes and monocytes. Lymphocytes are small and round cells, hyperchromatic nucleus. T cells are derived from thymus and B cells from bursa of Fabricius, also found in bone marrow. Um, Monocytes are bean-shaped indented nucleus. Uh, it becomes mobile macrophages in tissues. It becomes mobile macrophages when it gets out the tissue, and it means big eater. Macrophages are epithelioid macrophages, less phagocytic, specialized in secreting cytokines fused to form multinucleated giant cells. Um, there are kinds of multinucleated giant cells, which are the Langhans, the nuclei at periphery. Next is the foreign body, is nuclei scattered throughout the cytoplasm. Tuon is nuclei at the center. Anisocytosis is abnormality in cell size. Poikilocytosis is abnormality in cell shape. Next is the fixed macrophages in tissues. These are macrophages that we can find in our organs. And this image is a sample of the process of platelet plug. Next is blood circulation factors. Factors that govern circulation of blood through tissue, cardiac contraction, changes in the diameter of the lumen of the blood vessels, and amount of blood. Forms of circulatory disturbances. First is hyperemia. It means increase of flow of arterial blood. There are two forms of hyperemia: the active hyperemia that increase the flow of arterioles and capillaries. Passive hyperemia can result to congestion, increase of flow in venous. Congestion is the increase of flow of venous blood. Ischemia, local anemia, decrease amount of blood in the organ or a region. This image represents the hyperemia and congestion. Next is circulatory disturbances, hemorrhage, escape of blood from ruptured blood vessels. And here are the classification of hemorrhage. Pectic A is less than 1 millimeter. Ecchymosis is more than 1 millimeter. Agonal is the size of pectial and ecchymotic hemorrhages. The image shows pectic A from the upper and ecchymosis from the lower. Suffusion is diffused, flat, often irregular shaped areas of humor. Hematosis or hematoma is spherical shape, extravasted blood mass. Purpura is disseminated extravascular hemorrhage. Circulatory disturbances, number 5 is sludged blood, an adherence of blood cells to each other because they are coated with the film of plasma proteins. 6 is thrombosis, clotting of circulating blood on the wall of blood vessel. Next is embolosis, is condition formed once a thrombose becomes dislodged from wall of blood vessel and becomes a free lumen. Infraction, area of coagulative necrosis resulting from sudden stoppage of blood flow through an artery or vein. Edema is the accumulation of fluid in interstitial tissue from arterial end of the capillaries. And lastly, the shock, a circulatory deficiency characterized by the decrease in total blood volume, decreased volume of flow, and hemoconcentration.
Okay. So for the fifth and the last topic in our discussion is the neoplasia. So what is a neoplasia? So a neoplasia is a Latin term for swelling. It is also known as a tumor. So it is the formation of new cell or tissue type with atypical cellular structures and bizarre mitotic figures. So take in mind that all tumors are swelling, but not all swelling are tumors. So how does that happen? So there are swellings that are tumor-like but are not tumors. So there are 5. So first we have the cold abscess. So it is a slowly growing and developing localized collection in a cavity formed by disintegration of tissues. So in short, ito ay nana. So saan to usually nakikita? Sa tigyawat. So next we have infectious granulomas. So it is the nodular masses of granulation tissue composed of actively growing fibroblasts and capillary buds with macrophages and mononuclears, which are the lymphocytes. Next, we have the hematoma or hematocyst. So they are spherical-shaped hemorrhages. So saan ba nanggagaling yung hemorrhages? So it is from a damaged blood vessel. So next, we have the parasitic nodules, which are caused by nodular worms. So these nodular worms are Esophagostomum species. So they are usually found in the large intestine. So specifically, the Esophagostomum columbianum is found in the large intestine of sheep, goat, antelope, and camel, while the Esophagostomum stephanostomum is found in the large intestine of the monkeys. So last, we have the masses of intra-abdominal fat necrosis. So it is due to the reaction of lipase on fatty tissue of the abdomen, which results to saponification, and this is actually common in cattle. Okay. So next we have the histogenic origin of tumors. We have 2. So first we have the epithelial tumor. So if it is from the name itself, from the epithelial layer of the organ. So its suffixes ends with oma or carcinoma. So next we have the non-epithelial or mesenchymal tumor. So from the mesenchymal component of the organ, or it is the connective tissue component of the organ. So it ends with the suffix sarcoma. Sorry. Okay. So next we have the gross appearance of tumors. So they vary in size. So may mga factors na nag-influence sa size, which are the type of tumor, the blood supply rate of growth, age, and shape. So the question is, why does neoplasia happen? So neoplasia actually happens due to the mutations that result from the uncontrolled growth of cells that leads to the formation of neoplastic tissues. So these are the causes of tumors. So first we have the fetal risk, which is from the remnant of the embryonic tissue. So what is an embryonic tissue? Yun yung, kunyari, di ba, naging fetus tayo, naging baby, ganyan. So yung mga hindi nag-develop, nag-evolve. So the embryonic tissue is a remnant from when we were fetuses. So this tissue, if not removed or did not evolve at all, it will multiply rapidly in adult animals. So the more totipotent it is, the higher the mitotic figure and activity will happen. So next we have constant irritation. So constant irritation from friction, which will lead to abnormality in the cell organization. So due to friction, there is a formation of new cell type with high mitotic activity that leads to the abnormality of cell organization of tissue. So this leads to the dysplasia, neoplasia, as well as hyperplasia. So next we have the recurring infection. So as we all know, viral infections can alter viruses in general can alter the genes and chromosomes of infected animals that will result to atypical mitotic figures. So retroviruses actually can induce tumor formation such as lymphoid leukosis and tumor lesions. Next. Okay, so next we have the predisposing factors for tumors. So first we have age. So age is from the impaired ability of cells to eliminate the free radicals. So it usually occurs in older animals. So the second is the color. So pag absent ng melanin, so if you have limited or your body does not produce melanin, so mas prone ka magka-cancer. Why? Because melanin protects us from the UV radiation from the sun. So next we have the heredity. Okay, so for the color, so yung usual na sakit doon is the squamous cell carcinoma. So it is common in white-skinned animals. So yung mga albino, ganun. Okay, so third is the heredity. So it is the aberrant genes and breed predilection. So in genetics, there is deleterious genes. So yun yung may sakit na genes that will and may run in the family. Okay, so next, the fourth one is exposure. Exposure from environmental contaminants or mutagens. So what are mutagens? So mutagens are cancer-causing chemicals. Some are actually found in the food such as nitrates and nitrites. These are used for food preservation. Some other contaminants are also found in the air, especially nowadays, now that air pollution is a problem. So next we have species predisposition. So for household pets, so for dogs are actually prone to mast cell tumor and cutaneous histiocytoma, while cats are only prone to mast cell tumor. Next, we have the body site predilection. So, how does this occur? So, tumor grows on specific body types. So, for example, we have plastocytoma. So, it only grows on the digits, face, mouth, and ear canals of dogs. And then we have squamous cell carcinoma. So, head, eye, ocular gland, or the genitals of the horses. And then we have osteosarcoma. So, from the name itself, osteo, from the bones, the long bones. So it is common sa mga large dog breeds, and I think I saw one osteosarcoma case in the clinic sa Dalmatian ata. Okay, next. So these are the principal mechanisms involved in neoplasia. So first we have genetic misprogramming. So it is the progressive misprogramming of genes, of gene function at any step of differentiation or the selective reactivation of genes involved in early embryonic development. So next we have the uncontrolled mitosis. So due to the lack of mitotic activity, as cells become unresponsive to the controlling mechanism which results to neoplasia. So sa uncontrolled mitosis, actually, cells gain immortality and they become unrestrained and uncontrolled at the same time. Okay. Next are abnormal cell surfaces. So first we have marked by decreased adhesiveness. So it is a result from a major defect of cell surface adhesion molecules. So next we have invasion, which is a manifestation of the capacity of the malignant cells to spread by invasion of soft tissues, lymphatics, and blood vessels. Next we have implantation on new surfaces. So it is growing along the neoplasia growing along the serosal surfaces, forming a velvety carpet throughout the body cavity. And then we have metastasis. So metastasis to new sites through lymphatics or blood vessels. I'll be discussing more of metastasis later. Okay, so more of the principal mechanisms involved in neoplasia. So next we have the dysfunctional cytoskeleton. So it is the abnormal microfilaments and microtubules. So what do I mean by that? So there is an aberrant cell shape, aberrant movement, aberrant chromosomal stability, and usually this is due to the abnormal polymerization of tubulin. Next we have the failure of cell-to-cell adhesion. So gap junctions are the communication between cells. But in this case, it is broken and it prevents the direct communication between cells. And therefore, the metabolites are not shared accordingly and properly. Also, the intercellular junctions become abnormal. Okay. So next, the metabolism is shifted. So the regulation and flexibility of protein synthesis are lost. And then we have angiogenesis and fibroplasia. Okay, so what is the difference of these two? So angiogenesis is the formation of new blood vessels as capillary buds, while fibroplasia is the formation and proliferation of fibrous tissue. Okay, so we have two forms of neoplasia. So first is the benign. So it is the localized formation of ulit. So next we have the two forms of neoplasia. So first we have the benign. So it is the localized formation of mass. So there is a presence of capsule which contains homogeneous content. So isa lang yung laman niya with one color only. So it is potentially and generally harmless unless it is interfering with a organ function. I'll be discussing it later. So there is a slow and discrete growth. There is no metastasis. And it may be pedunculated or meron siyang tangkay kumbaga. So there is no infiltration to surrounding tissues due to the presence of the capsule. There is a prominent cell differentiation and its terminology ends with the suffix "oma" only. Okay, so for the generally harmless part. So if yung benign neoplasia or tumor grew sa may, let's say sa may renal tubule, renal artery. So as we all know, the renal artery supplies blood to the kidneys. So if it grew there, ang mangyayari is there will be ischemia. So pag nag-progress yung ischemia, pwedeng magkaroon ng kidney failure kasi wala nang na deliver na blood, oxygenated blood dun sa kidney. Okay, so that's an example. So next we have the malignant. So malignant, as we all know, is the cancerous one. So anaplasia and metaplasia occurs. So for the anaplasia, so there is an enlarged nucleoli, there is hyperchromatism, there is increased mitotic figure, there is atypical mitotic figure. So for the metaplasia, so from our previous topics, it is the transformation from one tissue type to another tissue type. And then there is a non-homogeneous content. So heterogeneous naman siya. So medyo madami, iba-iba yung laman niya. So there may be pus, there may be blood, and so on and so forth. So there is ulceration and hemorrhaging. There is lymphatic and blood vessel invasion. And there is many mitotic figures. So transplantation occurs. So what is transplantation? So transplantation, yung pagtubo niya sa distant or malayong tissue. So how does it occur? So since there is lymphatic and blood vessel invasion, so dun sila dumadaan. So can and will recur. So dahil nga kumakalat sila, ayan, so mag-recur siya kahit alisin mo siya. So their terminologies ends with the suffixes carcinoma or sarcoma. Okay. Oh. Okay. So, last line. Okay. So, for the manifestations and defects in anaplasia, so we have anisocytosis. So, it is the abnormality in cell size. So, next we have the increased mitotic index. Then we have metabolic shifts, which is the estrogen and androgen receptor damage. Next, we have the secretion of abnormal proteins. So what are these abnormal proteins? So we have alpha fetoproteins, prostatic specific antigen, which is specifically in men. So when there is a high PSA or prostatic specific antigen, so prostate gland hypertrophy may progress into becoming prostate cancer. And then we have interleukin 6 or IL-6. So it causes renal cell carcinoma. And then we have the collagenase and elastase. So with these two together, so it actually destroys the connective tissue kasi our connective tissues contain collagen and elastin. So these enzymes destroy those two. So that is it for my discussion. So again, yun lang. Thank you, Doc. Thank you for listening. Yun lang.
Thank you.
Thank you, Doc.